Okay, I will do about your introduction. So everyone, you are welcome to this Ecuadendro 2025 conference, the first Ecuadorian on dendrochronology topics. Good morning, good afternoon, and good night. Say yesterday the Dr. Nail. So the Professor Dr. Mahmuda Islam comes from Department of Forestry and Environmental Science, Cellular University of Science and Technology. About his biography, the Dr. Mahmuda Islam earned her PhD in Natural Science from the Institute of Geography of the University of Erlangen-Nuremberg from Germany. She is a professor at the Cellular University of Science and Technology in Bangladesh, and she is currently completing a postdoctoral position at Washington State University. Her research focuses on understanding the factors influencing forestry growth and wood anatomy in relationship to climate change. The Dr. Islam aims to develop solutions to mitigate the impacts of climate change on forests by combining dendrochronology, wood anatomy, isotopic analysis, and geographical perspective. She works to improve forest management practice through the study of terrain wood anatomy, enhancing the sustainability of forest ecosystem. The topic that she will speak in this Ecuadendro 2025 conference is about cellular dendrochronology in tropics, current status, and future prospects. So you are welcome to start there, Dr. Mahmuda Islam, and we are very honored to share this talk with you and with us.
Okay, thank you so much. Are you watching my slides?
Yes, it's possible to see you and hear you.
Okay. Hello, everyone. Thank you for joining my talk. I'm really happy to give a talk here in a first Ecuadorian Dendrochronology Conference. And thanks to Kelly. Today, I'm going to talk about the current status and future prospects of cellular dendrochronology in tropics. Cellular dendrochronology. Before that, I have to say, what is dendrochronology? Especially dendrochronology deals with tree rings. Dendro means the study of, dendro means the tree, and logy, study of. And then the dendrochronology, especially the time series analysis of tree ring. And cellular dendrochronology mostly deals with the time series of wood anatomical parameters. Now, tropical forest and cellular dendrochronology, how these two are related. Tropical forest comprise of nearly half of the world forest cover, and its response to climate variability have important consequences of global carbon cycle. Tropical tree growth, physiology, hydraulic behavior affected by climate change. These all the phenomena, all the concept encode the tree ring, and tree record long-term historical growth and physiological information in rings. And ring analysis means dendrochronology, tree ring analysis. And xylem characteristics, especially wood anatomy, are very suitable or substantially controlled by the environment. If there is the environment is good, then the growth is good. Ultimately, the wood anatomy behaved with the environment or climate. Then tree ring study, globally tree ring study, this map shows the distribution of tree ring study sites worldwide. The scale bar, dark blue, indicates the 500 publications per country, and white indicates no studies at all. Look at the tropical parts. Before the 1999 and before that period, there is no study in tropical regions, which indicate that treating studies mainly concerned in the Northern Hemisphere, USA, Canada, and Europe. A very few treating studies in the tropical and subtropical region. However, during the past 2 decades, tropical dendrochronology developed rapidly after the review of Martin Obbs in 2022, highlighting the potential of tropical dendrochronology, and then the review of Rosendahl and Zodema in 2021, and Brennan and colleagues bring this rapidly growing science informed and shown its application in various fields of ecology, climatology, archaeology, and physiology. Some other recent review clearly showed how rapidly treating science in tropics developed. These are the very interesting Brennan and Zodema, all other authors, they did the rapid development of treating study in tropics. These are the trend. Also, lastly, we reviewed one paper. They showed the trends in tree growth and intrinsic water use efficiency in the tropic under elevated CO2 and climate change. This is another paper like tropical and subtropical dendrochronology, treating study, dendrochronological advances in tropical and subtropical research, dendrochronology in tropics, and especially in pinus species. These all the study, when accumulate all studied in tropical region, most recent review on pantropical treating studies presented 459 studies in tropical and subtropical region. However, cellular dendrochronology, that is the studies on the time series of wood anatomical parameters, have not been flourished in such a space. So far, one of the pioneer tropical studies on vessel chronology of peak, published in 1999 by Puminongjong and Park in Thailand. Then in 2007, there is a long gap, but precipitation was reconstructed from 1743 AD from the early wood vessel chronology in South India. And this is a Bhattacharya first paper. And then recently some chronologies of wood anatomical features, mainly vessels, were developed in Mexico, Peru, Bhutan, Himalayan. They did some cellular dendrochronology in tropics. Even in temperate region, cellular dendrochronology was successfully applied to a limited number of species, including oak, beech, chestnut, and several pine species. Temperate also limited in some species, not so many species in temperate region. Why, now come to the point, why cellular dendrochronology is not developed like the other science developed rapidly? It faces so many difficulties. Like main challenges in wood anatomical time series developed in tropics, like a time consuming. Cellular dendrochronology is a time consuming process, and it takes a lot of time to process the data, to develop the cellular chronology. Lack of relevant instruments in many institutions. Lack of required image analysis software. Lack of skill required to prepare the thin sections and measurement. Lack of funds. But there is a huge potential tropical forest in tropical region. Tropical regions are rich in various forest types, including tropical evergreen forest, tropical mixed semi-deciduous forest, tropical deciduous forest, mangrove forest, freshwater swamp forest, and village forest. they have a rich biodiversity. Tropical forests are high in tree diversity, nearly 50,000 tree species, and many of those tree species form growth ring boundaries. Some are explored, some are yet to be explored. A preliminary study we have done on the wood anatomical structure on 27 species was conducted from a moist tropical forest of Bangladesh. Based on the growth of tree ring boundary characteristics observed in thin section, revealed that around 90% of the species have distinct growth ring boundaries. 90% species. Evergreen 8 evergreen, 15 deciduous, brevideciduous 4. They form the distinct growth ring boundaries. Out of 27 tree species, 14 species form fairly distinct growth ring boundaries. These are delineated by marginal parenchyma. 13 species form distinct and indistinct growth ring boundaries demarked by thick wall latewood fiber or fiber zone. Cluster dendrogram represent these 27 species into 5 clusters, and these 5 clusters, first 3 clusters is mainly marginal parenchyma. That means most of the tree species as in tropical regions, they formed ring boundary by using marginal parenchyma and determined by growth ring boundaries in tropical forest, most tropical forest. For example, Cicracia tabularis, Tuna ciliata, Lagerstomia speciosa, these 3 species wood anatomy, here we present these 3 species wood anatomical structure. Like first one, they form, this is the, this one, we'll look at the image and see, they formed a clear growth ring boundary and early wood basal. They have also the early wood basal. Tuna ciliata is a ring porous and Lagerstomia speciosa are ring porous species. But Cicracia tabularis is a diffuse porous species. Basal, and by using these anatomical characteristics or basal characteristics, we develop basal chronologies of Cicracia ring porous, Cicracia tabularis, and 90 years basal chronologies from 1925 to 2015. Also vessel density index, total vessel area index, mean vessel area index. These are the chronologies developed from the Cicracia tabularis. And after making this chronology, we test the climate sensitivity of wood anatomical parameters. Then climate sensitivity of basal chronologies to climate was tested. Mean minimum temperature had the stronger influence on basal parameters. Temperature had the negative influence. Ring width and number of vessel. Higher temperature increased the vessel density. Look at this one. Temperature mean has a negative influence on the vessel parameters, especially ring width and other special parameters. Vessel density increased with temperature. Mean vessel area has an important role in. Another study revealed the climate sensitivity wood anatomical features of 2 ring porous species, which are different in their life strategies and ecological guild. Tuna ciliata is a semi-ring porous, shade-tolerant pioneer species, low density, fast growing. Lagerstomia speciosa, ring porous, shade-tolerant, high wood density, slow growing, growing low-lying and land. Ring porous species and large early wood basal, smaller late wood basal. And we found the larger early wood and smaller late wood basal. Several basals variables were measured and chronologies were developed, and we found some correlation with temperature and precipitation, especially in spring temperature, March, April, May. have a negative influence on vessel parameter and ring width. Another cellular dendrochronological method used to measure the xylem hydraulic conductivity. By measuring the cell lumen area, mean vessel area, or tangential length and radial length, we can measure hydraulic conductivity. And xylem hydraulic conductivity also important parameter in cellular dendrochronology. Xylem hydraulic conductivity chronology in 3 species like Cicratia tabularis, Tona ciliata, and Lagerstomia speciosa. And hydraulic conductivity, strong effect. This climate also strong effect on especially Cicratia tabularis conductivity, and especially BPD, vapor pressure deficit, has a positive influence on the Cicratia tabularis, but Tona ciliata and other Lagerstomia speciosa, the temperature mean is important factor, especially pre-monsoon temperature, importance for Lagerstomia speciosa. Strong and Tona ciliata temperature, minimum temperature, negatively influence on ring width and vessel parameters. Also precipitation and other parameters. However, mean vessel area plays an important role in hydraulic conductivity. Hydraulic conductivity is an indicator of hydraulic efficiency. Higher mean vessel area, look at this one, higher mean vessel area leads to higher hydraulic conductivity. Higher hydraulic conductivity means higher hydraulic efficiency, then vulnerability is also high. On the other hand, higher vessel density is an indicator of safety mechanism. Higher vessel density ensures that some vessels remain functional when others affected by hydraulic failure during the drought-induced water stress and other extreme events. Nevertheless, there is a trade-off between hydraulic efficiency and safety. When conductivity is high, then vessel density is low. This is the inverse relationship. This inverse relation was stronger when analyzed on the tree community level than on the species level. Vessel density in x-axis, there is a vessel density. In y-axis, there is a diameter. DH means hydraulically weighted diameter. And in this case, we found Cicratia tabularis, there is these 3 lines represent 3 species. Gray one represents Cicratia tabularis, orange one represents Tona ciliata, and blue one represents Lagerstomia speciosa. And overall relationship is higher. That means when vessel density is more, the diameter is small. That means if there is when there is a drought or when there is any extreme events, the number of vessels is more, but the diameter, they make the small diameter vessel. This is the one of the adjustment mechanism of tropical tree species. And it's called the vascular efficiency and vascular safety. It's a safety trade-off. It's called the trade-off between hydraulic efficiency and safety. By using, also we tested this trade-off, safety and efficiency, in other species, Albizia procera. The vessel features and hydraulic trade chronology was developed in for this species, and very strong inverse correlation between vessel density and xylem vulnerability curve. And also another species for teak, Tectona grandis, we developed 56 years of long first row vessel chronology. First row vessel, early wood vessel, also encode the climatic signal. And this is the first row vessel with climatic signal. Pre-monsoon nighttime temperature, especially nighttime temperature mean favor the xylem cell formation and hence increase the hydraulically weighted diameter. However, during the post-monsoon season, an increase in temperature maximum limited further differentiation and enlargement of vessel, thus reduce the DH. Higher precipitation reduced latewood growth, but favored vessel formation enlargement mainly in the monsoon and post-monsoon season. By using cellular dendrochronology, we also reconstruct extreme events like drought and flood. Also, we can correlate drought events, flood events by using cellular dendrochronology. In Bangladesh, there is several drought years and flood years. We tested two drought years, 1999 and 2006. In comparison of the normal year, the drought year had longer dry season, short growing season, low precipitation, higher minimum maximum temperature. How these drought years reflect on the tree rings, like especially in cellular dendrochronology. In 1999, the vessel size is small, that means diameter is small, and
Hola, Dr. Mahmuda.
sorry for okay. I'm
Okay.
doing sorry.
You can
Okay.
continue.
Okay. In drought year 2006 and 1999, ringweed 54% ringweed reduced, and number of vessel 45% vessel reduced, total vessel area minus 44%, vessel density 2.3% vessel density. And also it matches with SPEI, Standard Precipitation Evaporation Index. The drought also affected the ringweed and also the vessel chronologies, number of vessel, mean vessel area, and ringweed. Special correlation of, also we did some special correlation and we found a special correlation with them. Like in Bangladesh, the red, red more negatively correlated with temperature and especially linked with the SPEI. Flood event, we have a lot of flood event in Bangladesh. Even the South Asia have a lot of flood events. We can use cellular dendrochronology, also the isotopes in to reconstruct the flood events, even to see how previous flood events impacted on the tree growth or cell level growth. Three major flood years in Bangladesh, 1974, 1988, and 1999. And we found these three flood years affect our tree growth, especially vessel growth or cell level growth. Like in these three flood year, we found the smallest ring width and also vessel density is more, diameter is small. These three flood events even match with our oxygen isotope results. That means oxygen isotope also low in that case. Mean vessel area, vessel density is high, but total vessel area is low in these three flood events. And these flood signals also match with the rivers, river water, near the one forest site, near one river level, Monu river level, match with the correlate with vessel density. This flood signal also you can see in the one paper, our one paper. There is a flood signal and cellular wood anatomical parameters in Lagerstomia speciosa in Bangladesh. Now, future prospect. We have more species, many more application of cellular dendrochronology in tropics. We have a lot of species. We have a lot of diversity of species. They form the distinct growing boundaries, and we can use cellular dendrochronology in tropics more. Last, I want to thank my Professor Achenbroning, also my university, Sutherland University of Science and Technology. I especially thanks to my all co-authors, special thanks to my one authors, Mizanur Rahman, and also Kelly, organizing such a nice conference. I have, if you have any questions, it's welcome. Thank you so much, everyone, for listening my talk.
Thank you so much, dear Dr. Mahmuda Islam. Very interesting your talk. We know that the importance about the cellular issues when we are working about dendrochronology project is very important, very interesting to know about that. And all your research with your scientific teams are a great job that you are doing. And thank you so much. And we have a question from Tanvir. And Tanvir, you are welcome. You can make your question, please.
Thank you, Kelly. Salam, madam.
Yeah, thank you.
Salam, madam.
Yeah, I am here.
Okay, madam. Thank you so much. Madam, I'm Dr. Tanvir from Pakistan Forest Institute. You have
nice
presented very nice. Thank you so much. Thank you so much. You have presented very nice and very informative presentation in your this session. I have just joined a few minutes ago, and I think I have you. I think you have mentioned these things are not. I don't know, but I have a question. How you tackle like it's easy to tackle ring porous wood, but how you tackle these semi-ring porous and diffuse porous tree ring in your species? I have very short question.
Okay, thank you so much. Thank you for the nice questions. Yeah, it's difficult for it's difficult to diffuse for a species, but every species we have found some pattern, like it's growing like a early wood is a big size, some big size. It's a gradually increase the size of the cell or vessel. That means there we found in our some species like not ring porous, diffuse porous, but they have some pattern, like a fiber wall thickness is going slowly. That means going decreases. Also, the size is going decreases. We found in our species some pattern, and by this way you can measure the early wood and late wood. Also, you can measure vessel density, vessel diameter.
But madam, I think when you have a clear demarcation for studying dendrochronology, I mean to say what is the age and what are the events. So I have no very, you can say, grip on this subject, but I have just want to know how to take it. And another, madam, question, what software you have used in your study? I mean to say WinCell or something else.
Yeah, I use in my cases, we use the WinCell software. We have WinCell software. We are using WinCell software for measuring the lumen area and all the vessel parameters.
Okay, madam. Thank you so much. Thanks a lot.
Okay. Thank you so much.
Thank you so much, dear Tanvir. Maybe another participant, would you like to make a question? You are welcome, please. Okay, I have a question. I am
Okay.
curious.
Okay, thank you.
I was reading different manuscript that when different researchers are working with anatomical feature or with anatomical parameter, they choose to work with an average vessel lumen area, how Tember say, in vessel lumen area, total vessel lumen area, in true ring temporary series. With these 4 variables, is enough for achieve of this kind of this project about what anatomical research, or we need more variable about when we are working on with anatomical description in
Thank you.
any topic there.
Okay, thank you, Kelly. Yeah, it's a good to have more anatomical parameters. Yeah, but if you have some parameters, but using some equation, you can measure other parameters. Like if you have the mean vessel area, total vessel area, vessel density, then you can measure hydraulic conductivity, you can measure vulnerability index, you can measure other parameter by using those parameters. But if you measure the cell wall thickness, it's a good idea because cell wall thickness influenced by climate very much. Then if you can measure cell wall thickness even, then it's a very interesting. You can add this parameter. But if you have more parameter, it's more useful to measure to predict the climate, to predict the environmental condition.
Very interesting. Very interesting. And I can understand that when you are working with this topic about know the relationship or the interaction that there are with the climate information or the climate data with the vessel features is a great job, is a big job, and you face a lot of challenge because make this measure the ring by ring.
Yeah, annual, yeah.
Working with all these anatomical parameters is a big work. I can understand you how it's work. So maybe another participant have any question? You are welcome. Okay. Dear Dr. Mahmuda Islam, thank you so much for this opportunity that you give us.
Thank you so much, everyone. Yeah.
This is so interesting and nice because the way of the science, especially in dendrochronology, is very important. And thank you so much.
Thank you so much for giving me a chance to deliver my lecture, and also thank you for everyone join in my talk. Thank you. Thank you so much.
So we will have our break time.